In 1947, a virus was isolated for the first time from rhesus monkeys residing in a forest in Uganda. It was later named the Zika virus. The first recognized case of Zika virus infection was in Nigeria in 1953. However, it wasn't until the 2007 outbreak in the state of Yap that Zika virus came to the public attention. The Zika virus stems from two lineages, the African and the Asian, with the Asian lineage being better known due to significantly more cases than the African lineage. Neurological and congenital complications have been associated with the Asian lineage of the Zika virus. And today, we follow the life of Zika viruses Flavia and Arbo.
Hi, my name's Flavia and this is my brother Arbo.
Hello.
We are the Zika twins and belong to the Flaviviridae family of the Flavivirus genus. We are classified as an arbovirus, meaning that we are transmitted via arthropod vectors.
Our favorite arthropods to be transmitted by is the mosquito, and our favorite mosquito species to hitch a ride with is Aedes aegypti.
The Zika virus originally was transmitted through a salvatic transmission cycle, where the virus was passed from non-human primates to humans. In urban areas, especially in areas where currently Zika virus is considered endemic, the Zika virus has had a human-mosquito-human transmission cycle.
Hi, I'm Aedes. I am commonly found in urban areas. I am a friend of Flavia and Arbo. They're hitching a ride with me right now.
Being a Zika virus can be so tiring, so during our time in a mosquito friend, we like to incubate for 8 to 12 days. When in a new host, I like to take time to settle in and spend 3 to 12 days dormant before the host realizes I'm here.
I have been sick for a couple of days now. I have been experiencing myalgia, fever, edema, and I have experienced retroorbital pain, vomiting, headaches, and a rash. Could this be Zika virus?
The following are diagnostic tests that can be used to detect the Zika virus. Reverse transcriptase polymerase chain reaction, or serological detection of the Zika virus IgM via the ELISA method. However, IgM assay is not accurate as the nature of flavivirus cause cross-reactivity in the body's immune system due to the previous flavivirus infection. The gold standard in detecting other virus infections is to detect the viral nucleic acid. Reverse transcriptase PCR requires the patient's serum for the best possible detection of the virus. Limited data has shown that Zika can be detected in the saliva. However, a study done by Musa et al. Also showed that in some cases it can be detected in the serum but not in the saliva, and vice versa. Amniotic fluid samples have also been tested using reverse transcriptase PCR, and it is possible to detect Zika. However, the sensitivity of detecting it in the amniotic fluid is still unknown.
Recent reports have suggested that I cause microcephaly, even though the development of microcephaly is not well understood. So, let us seek the light of new knowledge and see what scientists know so far. Microcephaly is a nervous system disorder that causes a baby's brain to be smaller in size and not fully developed. Through neuroimaging and autopsy studies, scientists have found that in most cases of suspected Zika virus-induced microcephaly, there is calcification of brain tissue largely at the junction between the cortex and subcortical white matter. Other abnormalities found were ventriculomegaly and enlarged cyston magna, abnormal changes in the corpus callosum, and cortical malformations in the frontal lobe. Testing of maternal and fetal Zika viral loads found that the fetal brain contained the highest viral load. A study by Franca et al. Found a correlation between infection by Zika virus and microencephaly, with the development of a rash early in pregnancy in Zika virus-infected mothers related to a smaller mean head circumference of the infant at birth. Ultrasounds done on mothers with suspected Zika virus infection revealed the following in the fetus. The fetus had abnormal brain morphology characterized by cortical atrophy, mild ventriculomegaly, gross parenchymal calcifications, smooth cerebral surface with few cortical convolutions, and an increase in the subarachnoid space. Microscopic evaluation of the placenta from infected mothers postpartum also found intervillous thrombosis, villus infarction, and foci of dystrophic calcification without viral cytotoxic changes. The development of microcephaly could be an acquired immune response. Normally, there is a high amount of gangliosides, which is a type of glycolipid present in the brain tissue. The gangliosides are responsible for normal brain development. During viral replication of the Zika virus in the fetus, the virus may incorporate the host glycolipids and glycoproteins, including the gangliosides, in such a way so that they become antigenic in the fetus. This initiates an immune response in the fetus that reacts with the gangliosides expressed by neurons, causing damage to the development of the brain structures, resulting in microcephaly. Another theory is a microcephaly retinoic acid theory. Retinoic acid is derived from vitamin A and is taken up by the nucleus, where it binds to retinoic acid receptors, which is RAR and RXR, on the promoter region of some genes. RAR and RXR are important transcription factors influencing RARE consensus sequences in the promoter regions of genes, which are involved in neural tube development. RARE consensus sequence repeats have been found in the Zika genome, and the greater the number of repeats, the greater the virulence of the virus. It is theorized that when viral DNA is incorporated into the host DNA, regions of the viral DNA that match regulatory regions of particular host genes may then influence their expression. Having said that, I now wonder what Arbo is up to.
Zika RNA can be detected in the semen for up to 134 days post-infection, which could mean possible sexual transmission even after the resolution of symptoms. I am also detected more frequently in saliva within the first week of onset of symptoms than in blood. However, in some cases I can be found in blood, but not saliva. The first reported case of sexual transmission was in 2008 and involving an American scientist conducting fieldwork on mosquitoes in Senegal and his wife. He had experienced symptoms associated with Zika infection and had returned home. His wife, whom had not travelled outside America at that time, presented with symptoms also 2 weeks after having unprotected sexual intercourse. Testing of the husband's semen was not due at this time, however 10 weeks later the husband's sperm, along with blood and urine, was tested after he presented with hematospermia. Sperm and urine was positive for Zika virus, while blood was negative.
There is no knowledge on how Zika replicates in the urogenital tract of males, what cells are affected, and whether the virus still remains virulent within the urogenital tract. Only one report has spoken of virulent virus being cultured from infected sperm. Inadequate testing or no testing of sperm at the time of suspected sexual transmission means that possible sexual transmission can be confirmed without a doubt. There are various theories on the mechanisms that lead to the pathophysiology of the microcephaly, but none have been confirmed as the true mechanism. While adequate testing methods of the Zika virus exist, cross-reactivity within an individual's immune system with other flaviviruses can result in unclear results. There is a lot of emerging evidence to support the notion that Zika virus causes microcephaly. However, no theory mentioned has been proven to be the cause of the pathophysiology.
This presentation was brought to you by the University of Adelaide Neurological Diseases students Celine, Jacqui, Mirabel and Nick.
We hope you have enjoyed coming on our journey with us today. Take care, and don't forget to use that mosquito repellent, or we might just become new residents of your body.